Good afternoon, everybody. So, today I wanted to talk about integration between different data and how to find differences. Before I get to that, just to follow up something which we had, which I hadn't properly finished last time. Here I had made this, I showed you this re-implementation of UMAP in Python, which wasn't quite finished before. Now it is. So, and I've uploaded it here as a Python notebook. It's also, let me see if I find it here. I show it to you here from this screen instead of showing it live, and I can tell you roughly how it works. I think I showed this last time. I used this EF-NUGget data set, and what I did, I exported the PCA that we always had from Server and loaded it here in Python. So as a little hint, if you work with both Python and R using this H5 file format is quite useful. HDF5 is a file format where you can just dump a lot of matrices in, give them names, and then I can load them again. Like here I say, load the matrix that I have saved in this file under the name PCA. And because R and Python disagree in a row matrix or column matrix, so you have to transpose it. And what I then did is I first gave it to the standard UMAP function, which you have in R, and then setting these hyperparameters A and B to 1, that were the one in this, you might remember here, but we said last time when we went here through this, how far did we get in this? I think we went through that, right? Yes. So, yes, as you can see, we have here this neighborhood relationship thing. Sorry. Yeah, that's the one. I look with these parameters A and B. If you set them to 1, you just get the student T distribution, which is just a nice symmetric fat-tail distribution. And by setting A and B to something else, you can adjust the parameters a bit. And what I did in the end here, I decided to keep them to 1, just so that the formulas are easier, but it's no problem at all to add more complicated things. Then I let UMAP run fit this thing and give me here the result of the UMAP, and then I try to do it myself. And if you go through this here, what you can see is I find my 15 nearest neighbors, so I've always tried to copy the UMAP standard parameters. UMAP also works with 15 nearest neighbors. Then I do the similarity score, but we also talked last time where I let the distance exponentially decay from and so that the nearest neighbor has similarity score 1, and then it goes down exponentially with some decaying factor sigma, which they somehow put such that it gives log of where it adds up to a given constant. And here, in order to find the right sigma that causes this to add up, if you want to do this in Python, you have to do it like this here. I write myself here this function here, with exponentiation, and as you see, I have to, and here I put in a candidate value of sigma, and now I have a function where I can put in candidate values of sigma, and it calculates the sum that you here see, which is supposed to add up to log 2. So I want to find the root of this function here, and to find a root in Python, you use the root function from scientific Python's optimize function. So you just tell it, try to find this root function. You then have to give it some starting value where it should try first. And I think there I said try first with just the distance between closest and the second closest neighbor. And then you can then this way you get the sigma. So this is a typical thing that you often do. If you do something like this in practice, you have to use some root finding algorithm because it's a transcendental equation that you can only solve numerically. When we symmetrize this course, as we told last time, you might have forgotten the details, but you can look it up here, what we did here. And then what I did in the end is I compared the adjacency matrix I calculate this manner with the adjacency matrix that the software by the UMAP authors has done. And unfortunately, I didn't get exactly the same result, nearly the same result. I haven't found the bug yet. So whoever, if somebody feels like finding the bug in there, then you get a copy if you find it. And then I just, and then here the next thing we said that UMAP initializes by spectral clustering, while, so I do with spectral clustering as we did it last time. So I take my graph Laplacian, the adjacency matrix that we've just calculated. I transform it into a graph Laplacian by subtracting this degree graph. You might remember this from 3 weeks ago. I rescale it slightly so that it's not b minus a, but identity minus and b subtracted is multiplied in from both sides. And this gives you a scaled Laplacian, and the spectral clustering works better with the scaled Laplacian. And when I do this and take the first two, then I get these eigenvalues. And as you can see, the lowest eigenvalue is 0. We had this last time too, but we said the lowest eigenvalue of the graph Laplacian is always 0, and the corresponding eigenvector shows you the stationary distribution at the end of the diffusion process. But the next two ones are the eigenvectors or the patterns coming to the slowest decaying overall pattern, which corresponds to sort of the fundamental harmonic or the most slowly evolving and hence the most coarse-grained or global structure. And so I use the eigenvectors corresponding to these two, and of course this eigenvector has one value for each cell. So if I have two vectors, then I get an x and a y value. If I plot my cells, I get this here. For a comparison, these colors here are the same colors as this one. So as you can see, if you look at it, you basically see the fundamental ideas of these arms. You already see them, but they are sort of now put in together. And that's what I then put into the UMAP algorithm. And here I have implemented this UMAP algorithm. But you remember last time I said this is what we do. And now I put here the implemented simply this pseudocode. I've written it out here in Python. I think in the interest of time I don't go through it in detail, because I actually wanted to focus on something else, but now that I put all the effort into implementing it, I want to show it at least. And I run it, and if I run it, then I get a very similar UMAP as the previous one. Not exactly the same, so there seems to be lots of little subtle details that I haven't completely recreated from the software, but it's roughly the same. One odd thing is that in this UMAP thing, here for the repulsive gradient, you remember that was, or maybe you remember that was the repulsive term of this overall force potential. And the UMAP algorithm says, well, maybe if you want to, you might want to multiply your repulsive gradient with a factor to dampen it a bit or increase it a bit related to the attractive one. And by default, it's the factor 1, but I had to put the factor to 1/10 in order for it to work, 1/1. But here, the point of it is here you see a typical mathematical thing, and here you can see how this is when I'm translated into code. And you can notice the annoying fact that if you have a complicated paper and you try to re-implement what they do, usually you find out that it doesn't actually do the same, because there's some kind of small hyperparameter or things where maybe the paper and the software have drifted apart and do not exactly describe the same thing. So, the other thing I wanted to point out, you might, coming back to this a single cell thing. If we look at this paper, so this was what we did, what we started off last time, that we said this is our usual UMAP and this was the T-SNE. We wanted to know why is the T-SNE so different from the UMAP. And as you can now see, and to really go through this question, what makes the difference between the T-SNE and the UMAP? We can of course now here go through our mathematical text here and compare and see all the small differences and one major difference. The small differences are in the beginning, in the high dimensional space, we calculate the similarity scores, which here in T-SNE are done with this Gaussian kernel, E squared over sigma squared, and in UMAP, we use instead this kernel, which is exponentially decaying and doesn't have a square. Another difference might be here we just normalize the thing in a simple ad hoc manner to say that distance, the similarity to the closest neighbor is 1, while in T-SNE we say this thing should be interpretable as a probability density, and hence we have to normalize that such that all these values add up to 1. The UMAP people mention in their paper that they find it peculiar that this normalization doesn't really seem to be needed, and hence rightly mention that this casts doubt on this whole idea of interpreting this as a probability. And maybe you felt a bit confused when I explained this last week, when I talked about the stochastic event that point I chooses point J to be considered neighbor, where we make this a stochastic process of two vertices either being considered neighbor or not. A frequentist would say this sounds stupid. This is exactly what frequentists don't like about Bayesians, that something is either the case or it isn't. We don't want to assign probabilities to fixed stuff. A Bayesian probably wouldn't sound so happy about it either, because that's what kind of process is what's required. So it feels very ad hoc, this neighborhood embedding. And it's maybe one of these cases where you come up with an intuition, this idea of choosing neighborhoods first with the Gaussian kernel in the high dimension and the fat-tailed kernel in the low dimension. But then later you realize that this intuition led you to something which works, but actually the intuition is not that tenable once you try to really put it on a solid footing. And the point where things like this then become clear, that the intuition was to hand waving and that it really isn't a probability. Somebody else comes with an algorithm which does something similar, but for example says we don't even have to normalize to make it a probability. So it probably was never one from the beginning. So this was one difference in T-SNE and UMAP. The other difference was of course the low dimensional embedding. In T-SNE you remember we have this T distribution, the fat-tailed T distribution with parameter with 1 degree of freedom, and in UMAP we add these two extra hyperparameters A and B to be able to tweak the shape of these things a bit. And the third difference is how we do the optimization. T-SNE, we used this Barnes-Hutz algorithm. We imagine that it's the same like in physics where we do a particle dynamic simulation. We calculate the attractive or repulsive force between every pairs of cells and run there into the problem that this scales quadratically, and then we solve this issue by replacing all those cells which are far away from our currently treated cells by simply one big meta cell which is simply the center of mass of them all. And in this way we still exactly or more or less exactly solve our optimization problem. While in UMAP we do something else, namely instead of agglomerating a large number of cells which are far away from our current cells into one center of mass, we just pick 5 of them at random and say, as we picked at random, on average their center of mass should be where the real center of mass is, so we can use this as well. And in order to this be the same thing as before, we have to then scale this repulsion up from us having picked 5 to represent maybe 1,000 cells, so we have to multiply by this. And this is this last part which is missing in UMAP. And which strangely enough seems to be the main reason why UMAP and TSNE work so different. So all these small things that people focus on early, that I've mentioned before, probably don't matter how exactly you define the similarity score in the high dimensional, what shape you exactly use in the low dimension score, what kind of method for optimization you use, whether you use Barnes Hut or with rejection sampling. But what does matter is, of course, if the relative strength of repulsion to attraction differs by an order of magnitude, that makes all the difference. So it might be now interesting, and that was a bit my idea behind re-implementing these things, to see what happens if I now change the repulsive strength of UMAP to that we see in TSNE, or vice versa, to when the plots look the same. So that's what I wanted to say about this part. And now I want to come to the next question. I mentioned at the end of last time that in biology, especially in quantitative biology, we always want to compare. Biology, in a way, has these two modes of thinking. One is the descriptive mode of thinking, which is your classical idea that everybody has of 19th century biology, and that you also get from high school, where you dissect some animal and make lots of drawings, and then make a complicated description explaining that this flower has so many petals and so many sepals, and has a 5-fold symmetry, while that one has a 6-fold symmetry. So you just describe the big variety of shapes that you see in nature, or you describe the complex anatomy of a mammal by finding all the little anatomic details in the human body. But the other mode is, of course, the quantitative mode, where you measure something, and whenever you measure something, you get a number, you need something. This number doesn't help you anything if you can't compare it to something else. That's why all quantitative biology is always comparative. You always want to compare two things. In order to have a practical question where we do compare something, I thought I use this Schroeder integration. All this is hot. I'm looking for I worked a bit with this data set, and I want to show you something like this. So there will be a link to that somewhere in the web page. But I have rewritten most of the code here in my own R code. Unfortunately, it's so far not a nice Quarto document, but it's just code with comments in between, so it doesn't look as nice. But here I show you what it is. We take a data set that the server people have prepared in order to have something for us to work with. We call it the IFNB data set. IFNB, that stands for interferon beta. And so what they have is 14,000 PBMCs. You remember these are peripheral blood mononuclear cells, so just cells taken from the arm vein of some subject and then sorted with the fax machine to only keep our normal immune cells with only one nucleus, and pick out all the red blood cells and all the stranger immune cells with multiple nuclei. And we want to know how these cells react if you stimulate them with interferon. Interferon is one of these cytokines, of these messaging molecules that float in your body and that help you and that mark, in this case, alert about inflammation. So to explain this, everybody knows hormones, which are messenger chemicals that float through the whole body, but there's also more localized messenger, where you have for example, a place where cells notice that they've been infected or injured, and then each place produce alarm molecules which alert the neighboring immune cells that there's something happening. And one of these is interferon. And what these people here did is, you see, it comes from this paper here. And in this paper, they actually took samples from 6 different subjects and mixed them all together. And then they split the mixture in 2 halves. one half was stimulated by putting interferon onto it, the other half was not stimulated, and then they sequenced them separately. And in the beginning, for most of our talk today, we will not care about which cell is from which subject, but at the very end, we will realize that it actually would have been very important to care about it, and we'll try to pull out these things. That was a bit of the big point of this paper here was that usually, if you want to be able to tell afterwards which cell is from which subject, you would have to mark the cells somehow so that you can tell them apart afterwards. But they realized that, of course, if the cells are from different subjects, they have different genotype, they differ in their genetic material, so you will find difference in reads, and so you might just be able to tell whether two cells have been from the same subject or from different subjects by looking at the actual sequence of the reads. You remember, so far we always used the read sequence only to find out which gene it is from. But now we look how the read sequence differs from the reference sequence for this gene and might find that, that it differs in a specific manner, and we categorize all these differences that we find. And that will then allow us to say, okay, we seem to have 6 different ways how our reads can differ from the reference, and that's obviously because we have 6 different patients, and that allows us then to reassign which cell is from which patient. But I won't go into how they've done it. We will later simply use their table where they have already annotated each cell with the corresponding subject ID. For now, we just want to know how these cells, what happens to the cells when they get stimulated with interferon. And so, because they can later notice whether 2 cells come from different subjects or not, that we can see from the sequence of the reads, but we cannot see from the sequence of the reads whether the cells have been exposed to interferon or not. So we must not pool together these 2 things. We have to keep them separate and run them independently through the 10x single cell sequencing apparatus. And only afterwards we put them together. So what we have here is the Sura people made this Sura data package where they put example data sets. And I've now loaded this Sura example data set, and if you look into it, and let me start here, the fresh data set. What you now see here is what we have. We have here an active assay with counts and with already log normalized counts. And we have this STIM column, which tells us for each cell whether it's a control cell or a stimulated cell. And what Sura now wants is that we use Sura split command to split the count and data matrices into 2 submatrices, one for the control and one for the stimulated. I call this now interferon beta split. And now I run the usual standard analysis that I've always done on this. So I tell it to normalize the data, find the variable features, or find the 2,000 most variable genes, run the PCA, find the nearest neighbor graph, find the cluster, and then find the human. What I will then do is that I look at the plots, and because this takes a moment till it does that, I'll show you how these plots look like. Let's see. Okay. And once we do this, we will get a UMAP which looks like this here. And you see some strange symmetry. So the red cells are the control cells and the blue cells are the stimulated cells. And if you look at this, you notice how there is always this mirror symmetry between here, along this axis, and along this plane. You have a mirror symmetry. So what's happening here? What is happening is that cells which are of the same type, but they differ by being stimulated or not, they lift off from each other. So imagine you have some manifold which contains all the cells of some type, and let's say this, my phone is the manifold, and now let's imagine the stimulation causes these cells to change. And the way how the cells change is not within the manifold, but orthogonal to it, because the manifold only contains unstimulated cells in resting state. So of course it makes sense that if a cell senses interferon and switches to alert cell, that it switches to a state that the cell that is not present in our control cell. So we switch to a new manifold, which I now indicate by lifting up my phone a bit. And as you can see, I also flip my phone a bit. So let's imagine the lower end of my phone are cells which only change a bit to interferon stimulation, others which change a lot to interferon stimulation. And here you can see with these plots here where the two things seem to touch, here, here, and here. That must be cells which don't react much to interferon stimulation. These here and these here are the same cells which react strongly. Same way, this cell types also seems to react a lot between the two. Now, this is already from a published paper, so you remember a while ago we tried to identify all the different cell types, and that was a lot of work. We had to do clustering, like this here, and then we had to look at each cluster and find out which cell type it might be. And we had to check whether we made our clusters to fine-grained or to coarse-grained. Now the authors of this paper has already done it, and the Sera people included this in our interferon object. So we can see here the annotation from the paper. So for example, you see this blob here and that blob here, which are both two different types of monocytes. One the stimulated and one the unstimulated. So here you see the T cells, the helper T cells and the natural killer cells change a bit. And this orange one, which has such a strong effect, are the naive T cells. The activated have been activated before, so they probably don't change much. But imagine you had to do this analysis yourself. And this is really annoying because for each of these, because basically each cluster now appears twice, once for the stimulated and once for the unstimulated. And you have no guarantee that the borders between clusters will be properly in this, at the same place in stimulated and in unstimulated. So working with this clustering is really not productive. What we now want to do is to integrate the data, and by this we mean that we remove the differences which are caused by stimulation. So that for each cell here in the unstimulated, we try to find the corresponding stimulated cells which is closest to it, and then we move them on top of each other. So we find a new coordinate system, a new latent space to replace our PCA space, such that cells which are corresponding to each other in the two data sets will appear at the same place, even though one is stimulated and the other isn't stimulated. What we then can do once we have it is take one cell and a small neighborhood around this cell and say within this neighborhood, averaging over the 20-30 cells in this neighborhood, what's the difference between stimulated and control cells in this area? And because the cells have been put together, we then can get sort of for this place on the latent space an answer of which genes will go up if a cell here gets stimulated, and how does this differ from a cell here getting stimulated. And in that sense, we now have always two ways of looking at our cells. Either the cells still including the stimulant effect, or the effect of stimulation being regressed out. And over the time, people have discussed a lot of different ideas how to do that. The easiest idea, which actually works more or less, is the following. We do our PCA as we've done before, and we do it only using the red control cells. Because the PCA, and then of course with this PCA, we span a 30-dimensional space with a principal component which spans all the stuff that these cells vary. That spans all the dimensions along which these cells vary. And none of these dimensions will point in the direction into which the cells move due to stimulation, because there are no stimulated cells in the input to our PCA. If we then later take our stimulated cells and project them into this PCA space, then of course we will end up in the same space as before, only the stimulated cells, if we then later project the stimulated cells into the principal component space spanned by the PCA of the control cells, most of the stimulation effect will vanish. And this works sort of, but it doesn't work perfectly, because imagine the effect this here were to be the control cells, or the manifold folded by the control cells, and now my stimulated cells, they sit like that up there. So you have to now imagine that this line hovers sort of a few centimeters above the blackboard, while this line is on the blackboard. So if this is above the blackboard, and if I project it into the space in here, it sort of will work, but it will still be shifted, because any shift that is within this direction will still be there. And so people try to find better ways. And also, I want to describe two methods. One is called the mutual nearest neighbor method, where for each cell we find its nearest neighbor in the other set of cells. Maybe this cell has all these as nearest neighbors. And each of these cells have all these as nearest neighbors. And so we find for each cell the nearest neighbors among the cells in the other condition, and vice versa. And then we keep only those pairs of cells which are mutually nearest neighbor, which have found each other among their nearest neighbors of the other condition. And in that case, we get a bipartite graph where each cell where we have always connections, where every edge connects one stimulated with one control cell, and in that way makes a mapping between control and stimulated cells. What we can now do is that for these connected cells, we take the difference between these two cells in our latent space. So imagine beforehand we have done a PCA. Now this time we mentioned, I forgot to say. First we do a PCA on all the cells, not only in the control cells, but now in all the cells. Only this allows us to actually see nearest neighbors here. Because otherwise we don't have a distance measure, because if we don't have a common space for both cells, how would we judge distance? And then we find the mutual nearest neighbors. Now we have a bipartite graph connecting stimulated cells with the nearest control cells and vice versa. And we keep only the mutual axes which appear in both directions. And now in that way, we can now take for each pair of connected cells, we can take the difference between the principal component coordinates of these two cells, and can say this difference vector, that marks as the effect of the stimulation on a cell at this place in the latent space. And in this way, I now get a number of difference vectors which allow me to say, okay, these vectors, they tell me how much what I have to subtract from the cells here in order to get to the other space, in order to remove the stimulation effect. And in order to make this whole, make sure that we have a smooth transformation, what we do is that we do a kernel smoothing here. If we have here a stimulated cell and we want to find where it goes in the unstimulated cells, what we do is not that we simply subtract only this vector. Instead, we take a neighborhood among these cells and a neighborhood among those cells and find all the edges that connect any cell here with any cell here and average all these vectors in order to get the vector to subtract from the stimulated in order to get a corresponding position in the control cells manifold. And in order to make this averaging over neighboring cells, you can again do our kernel smoothing. We put around the cells, the two cells which are connected, we put a kernel, for example our tricube kernel again, which assigns to each cell to each cell a weight, depending when the weight becomes smaller and smaller, the further out we go, the further away we are from the center cell, and in that manner we get sort of a smooth amount of cells going down. I started to implement this, didn't get all the way through, but and also it's not what, so this is the first thing that I wanted to mention. Nearest neighbors, let's see if I find here. So this was the paper of 2018 which suggested this method that I've just described. Looking at these things, and the writing thing, and from the description that I've just seen you can already see that should be easy to implement if somebody wants to try. You can count this as a nice homework project, but it's a bit cumbersome because you have to be really careful to not mess up the numbering of your cells. But it should be possible. Now I want, a bit later, came from the authors of the Sörer paper, this paper here. Sörer integration. Let me see if I find the correct paper. Where is it? No. Scholar. Yeah. They came up with a slightly more involved thing, and that's what I now first want to run and show you how it works, show you what it does, and then I will briefly describe how it works. So first, what does it do? Let's see if I now this thing here should be run through. So remember, so far we have still here our not properly integrated data, like this stuff here, which we are not happy with. Here these things. I can also use sleepwalk. Remember that was this little tool that we did to always show you which cells are how closely related. And the way how I've done this here now is, you see I split the cells according to put all the red cells here on the left panel and all the blue cells in the right panel. But it still does the same as before. If I put my mouse under on a cell, then you see in green the nearby cells on either side. If I go, for example, here, then I can now see what is the corresponding cells in the other side. For example, you can see that this little corner here corresponds to that one. So this unstimulated cell probably becomes this stimulated cell under stimulation, while these ones here become this part. And these ones become this here. What you can also see, if you look at the color, of course, the cells on the opposite panel, which is not under my mouse, are a bit less dark green, because they are further away, because my distance contains the distance that the cells move in feature space due to stimulation. Here the difference is small, but here, for example, the difference is quite strong. It's dark green here, and on the right-hand panel, it's only light red. So let's keep in mind what we want to achieve is that we reduce this, that the distance to a cell of the other, the distance from a control cell to its closest stimulated cell should be no bigger than to its closest control cell. And to do this, what I now do is I run here this integrate layers function, which does an integration method which we call integration by canonical what is it? Correspond is the damn it. Canonical correlation. Canonical correlate. That's what it stands for. Correlation analysis. Canonical correlation analysis is a bit a weird thing. It's sort of a PCA that is split over two sets. In a normal principal component, we try to project our data into a space such that the maximum of variance is explained. In a CCA, we try to find two projection operators which project both of a vector of feature vectors into a common space, such we find that we get maximum projection, maximum correlation between the projected things. So what we try to do is, let's say we have a gene by feature matrix, or cell by gene matrix X and Y. So maybe this is controlled and this is stimulated. And the cell by gene matrix X and Y, we want to project it somewhere. Let's call this onto some smaller matrix. Let's call it X prime, and this one to some matrix Y prime by some suitable projection. And this now, in these two matrices, they are in a joint space, a d-dimension space, where d is again 20 or 30 dimensions as before. And what we want is that corresponding rows here should be matched to rows which are highly correlated with each other. And the way how you do, and now it gets a bit weird. What we really want to find, of course, is cells which correspond to each other. But that doesn't work because we don't know which cell we should correlate to which other cells. So what instead we want to find is genes that help us establishing correlation. So we try to find a linear combination of genes such that things are highly correlated. Let me try to do this. So I say I take here, so this is a cell by gene vector, and this here is a gene vector. So it gives weights to each gene, and in this way, this here makes us a linear combination of genes. So each cell is now described as a weighted sum of the genes with the weights given here. And no. Wrong. The cells. We give to each cell a weight, and then we average all the genes according to this, and then now I get a vector that damn it. I want to get into the gene. In the end, I need something which such that the genes correspond to each other exactly. For each gene, I make an average over all the cells with a proper weighting of the cells. So some cells might be weighted positively, other cells might be weighted negatively. And afterwards, what I get is what I get is a vector which gives us a value for each gene. And then I have another vector, y, tv. So this is now gene times cell, and again a cell vector. And now this is also the other way around, also gives us a vector which assigns a number to each gene. And in the middle, these two things meet, these two gene vectors, and we make a scalar product. Now, if we chose the cells such that the same genes get upweighted or downweighted, then this thing and this thing will be well correlated. So we say what we are looking for is the maximum is to find the vectors u and v that maximize this thing. And when we have found them, then we know that all those cells which have a high value in u are probably similar to all those cells which have a high value in v. And all those cells which are strongly negative in u are strongly negative in v. And that we use as the first coordinate in our Latin space. When we regress out all the u's and v's that we have, when we regress out this dependence, so that we can find another set of highly coordinated matrices. This is what we call CCA, canonical coordinate analysis. Problem is, we typically have many more cells than we have genes, at least reasonably variable genes, which will cause the effect that that this maximum will be degenerate. There will be many different ways of assigning weights to the genes in either of the two conditions, such that we produce two vectors which are perfectly correlated. And because of this, we can't do it that way. The standard approach would be to regularize the thing. That means to add some extra condition that we say we want to have these weights to be minimal. At the moment, we just want, in order to get a proper correlation, we make a side condition that UTX and VTY should just have unit norm, but we might do some more things that we, for example, say that the individual elements in U and V should be not too much. But again, we don't do that. The Sörder people write in their paper that they explored this, and it doesn't work because in that manner you get too many. If you get too many cells which have a weight of 0, and if all the cells have weight 0, then you can't match them up. So they decided to do a workaround, and that's the thing that got really confusing. I have to say, as myself, I read it many times, and I'm still not sure whether this is really the best way to explain it, how they go to consider it as a part of the CCA, because in the end, what they do is they make an SVD. SVD is a singular value decomposition. Everybody familiar with that? So singular value, just to write it down again, any matrix M can be decomposed as M is UDV dagger or V transpose with UTU like VTV is 1. So what we do is we take this matrix X, Y, T. So this is now a matrix of cells by cells, or cells in X by cells in Y, because the genes are in the middle. I think in the code I have written X transpose, so it will be the other way around. And for this I make an SVD by saying this should be UDV transposed. Now, to be easier to interpret what is going on here, I multiply this SVD from both sides by saying U transposed X, Y transposed V equals E. And now we went into the shape that we have here. What we've lost a bit is this side condition, and we have replaced it with that side condition. So while here we say these vectors, the individual vectors after the transposition should have L2 norm 1. Here we simply say the columns of U and V have this. And they makes again one of these hand-waving explanations of why this is. Either I haven't fully understood it, or it is hand-waving. Maybe another possible way to look at this is simply to say, okay, this might be a useful way of. You can look at it different ways to understand why this helps. But what we now do is we can now take the coordinates in U and D. I write this as U e to the 1/2, e to the 1/2. V e to the 1/2 T. So now this is simply a decomposition of a matrix in two smaller matrices, where this thing here is one thing and this thing is another thing. And as you can see, because I multiply this matrix D on the right-hand side, and the matrix D contains the singular values sorted by order, first the big ones and then the smaller ones. This matrix has columns which diminish in norm. The column to the very left has the highest norm and it gets smaller and smaller. So we can just keep only the top, the 10 leftmost columns here and maybe the 10 leftmost columns here and still have a good reconstruction of this whole thing. And what these matrices then have, you remember this matrix has one row for each cell and it has now these 10 columns that we decided to keep. So we give our cell now a 10-dimensional or 20-dimensional vector in a new latent space, which is the latent space in which these two matrices live. And this latent space is made such that if we calculate the correlation between all the coordinates, so for example, between the first coordinates over all the cells here and all the cells here, sorry, wrong way around. We see here that, yes, we have to see that part here. Let's just look at the code, what it does, and then we can see how we can try to understand. It might be easier to see what it's doing. So what I now do is, I run my CCA. Oh, looking at the code, try to remember this for a moment, what I've done here. Because what we've done here on my computer is we ran Seurat CCA integration. And what I wanted to show you is first how this result of Seurat CCA integration looks like, and then we will compare it with our SVD to see that this is sort of the same thing. So first to see what came out of our thing. You remember I ran this integrate layer function with the CCA integration function. I commented out here simply because it takes so long, and instead I will load the result. I mean, it doesn't take so long, it takes 5 minutes, but still I don't want to wait here 5 minutes. So I load this. Now I have to put these two layers again. You remember earlier we had split up the matrices into a control and a stimulated matrix. Now I put them together again, that I have one big one. And now I have this new EFNB. Now has several reductions. It has the PCA reduction that we used before, and now it has the CCA reduction, which is the reduction formed by these things here. So this is the CCA reduction for one, and this is the CCA reduction for the other. And if I do this now, and I can use this CCA interaction and say, using this CCA introduction, find again a clustering and find a UMAP such that the clustering is such and in this UMAP we should now then see that these things flip together, that this thing should end up on top of that one, these two things should sit on top, and these two things should sit on top. Because all those parts, all those aspects of our latent space which do not appear in both things, they cannot correlate, and we cannot map them into this space here. Because in normal PCA we try to find a representation which maximizes the variance, and now we have tried to maximize the covariance between this and that. Obviously, if you look closely, for this to be a proper covariance, what I should have done is subtract the mean from the matrices X and Y. So imagine for the moment that this thing is centered, that from each gene I have subtracted its average expression. So, and here you can now see how our integrated data looks like. Here is the stimulated and the control is below. It's a stupid way how Seurat plots this here, unfortunately, because it plots the cells in the order in which they are in the matrix. And so, of course, it first plots all the rod points and then it puts the blue points on top. In order to solve this, what I will now do is I take the UMAP that we just got from plotting and tell it add a color and so on, and if I plot this, I get the same issue now making the plot by hand. But what I can do is, I tell it here, randomly shuffle the arrangement of cells, like the orders of the rows in the matrix, and then now they sit on top of each other, and I can much clearer see that really all the regions of my UMAP now contain a mixture of stimulated and unstimulated cells. So it has worked to remove most of the effect of the stimulation. With that, I can now also look at clusters, which, and now if I do a clustering on this space, then of course each cluster simply contains a good mixture of stimulated and unstimulated cells. And if I now try to find cell types on these, then I find for each cell type an explanation how does the cell type look in stimulated and unstimulated. Obviously, this is a bit of work to do this, but here you can roughly see what we've seen as clustering before, as clusters before. I look at this one here. These are our natural clusters, and they agree quite well to also what the original authors of the paper have used, and then they managed to assign to each one a cell type. So now the cell type assignment worked much better after we have put them together. So, and yes, and now I want to compare this with what I do manually. So I take my 6,000 control and 7,000 stimulated cells, and I calculate here this cross correlation, this XTY. Or here I've written XYT on the board. On the screen I've written XYT because you remember in biology the matrices are always transposed for the statistics. So, and now I run my SVD. I simply calling this SVD function and telling it find the top 20 singular values. And if I do this, I have now here SVD $B. And here you can see how quickly these values are reduced. So it makes sense to keep only the first couple of them. And here we now see for each control cell, we get now 20 coordinates, and for each stimulated cell, we also get 20 coordinates. So this here are the 20 coordinates for the 6,500 control cells and for the 7,500 stimulated cells. And what I now do is I use this. I of course should multiply each control cell with the square root of D, as I've shown you here. I say my XYT is my UDVT. I have of course the U and the V, but I have this D in between, and I have to multiply the U matrix and the V matrix with the diagonal matrix in order to make sure that the rows properly go down, because at the moment in my U matrix each column has norm 1, but that's not appropriate. We know that the first components have a higher importance than the later components. So for this I have to multiply by the values to scale each column according to the singular value. And of course I have to take the square roots, because this D here contains the singular values for both matrices, so each matrix gets half, or square root of it. Now, what I can now do, I can again make this plot that we had before. You remember this one here, but this time I use the same UMAP as we have here, but I use our SVD for ins, and now you will see. Remember how here in this case the cells of the other side always are a bit further away. Here only a bit, here much further away. And if I now calculate the color, not from the PCA as before, but from these things, this problem will vanish. Now the same plot again, and this time the color is calculated from this U times square root of D or V times square root of D. And as you can see, now it works really nicely. Each cell has neighbors of the same distance in both the stimulated and the control. And we can now properly see which cells correspond to which other cells. And it's a bit of magic, but you can do this just with an SVD. So I do this now. I put these two matrices here below each other, and say this is the matrix for the control, this is stimulated. If I put them below each other and I have the matrix for all of them, this is why I make here the row bind. Make sure that my row names are still all in the right order, and I assign this as another reduction to my interferon D object. Now my interferon D object has lots of distractions. The PCA that we originally did, the CCA that Seurer did when we told Seurer to integrate it, and the SVD that we got when we tried to integrate ourselves. And we have a UMAP calculated from this PCA. We have a UMAP calculated from Seurer CCA, and now let's add a UMAP calculated from our SVD. I plot this again as before. I need to shuffle my cells, and we see again we get a nice agreement. Isn't exactly the same UMAP as what Seurer got, but it's reasonable. So for example, here, let's first look at this one. Here I can put the cell type annotations, and as you can see, it works. Each of the cell types from the authors corresponds to a blob here in my CCA. So my CCA seems to have worked reasonably well. If I take the clustering from on Seurer's CCA with my primitive SVD-based CCA, see, it also works. Only those clusters where the borders are not quite clear anyway, they seem to now be overlapped. And I can again use my sleepwalk tool to compare now the UMAP made from Seurer's CCA and the UMAP made from my SVD. And it works, seems, and as you can see, it seems to be quite similar. This big blob is here, it has this small thin protrusion, and this bigger blob, this thing here has this shape. So only here it's a bit weird. This thing might correspond to that thing, and this corresponds to something which that one doesn't really have a proper comparison. Now I keep saying my simple SVD because Seurer doesn't stop at the SVD. It then goes one step further. One thing they are worried a bit, but then they decide to ignore it anyway or show that it could be a concern, but in practice it isn't. That they say our SVD will only be able to map dimensions in latent space which appear in both sides. So cells which might differ from each other within stimulus, but they don't appear in the control cells, they will flop together. So for example, if you have one control type of cells, and this control type of cells, once it gets stimulated, it throws a coin, and with probability 50% it changes this way, and with probability 50% it changes in another way. Then of course this will confuse our CCA, because the variation between the two possible responses to the stimulus doesn't appear in the control setting. So we cannot get covariation because the variation doesn't appear on both sides. To check that, what we do is they perform a PCA on each of these things individually and check how much variation can the top 20 principal components explain, and how much variation do we capture if we project our data on this matrix here, on the space spanned by these here. And if we do that, sorry, actually on the space spanned by these here. And if we do that, we find that at least for this data set, the CCA seems to capture as much as the PCA captures. But we have a big advantage in this thing, because in this case here, our biology is, remember, in the experiment they took the cells from the patients and then they split them into two aliquots, stimulated one half interferon and not the other. So we know that the same types of cells with the same relative abundances appear in both samples. If we now, often you have another thing, maybe for example a typical case would be that people want to see use single cell sequencing to follow the healing of an infection. For example, you might use or a treatment of a tumor. You might have patients with blood cancer. You take blood cells from them before treatment and then after treatment. Now, this assumption that I just said, that the same types of cells are present in the same abundance, is now manifestly false. At least you would very much hope so, that the treatment caused the number of tumor cells to get reduced and the number of healthy cells to go up. And now the question is whether the CCA might cause whether this imbalance might cause the CCA to remap these things. And then they say what we now have to do is to make sure that the distribution of these U's in these V's are the same. Let me show you what I mean by this. Where have I put that? Hey, I thought lost that. So I use again this integrated CCAs here. So these are the this is the first coordinate for each cell, and let's here say EFNB $stim control. This is for the stimulated, and this is for the control cells. And I now make a density. And this is 1, and I make a density, a kernel density estimate of the other one. And as you can see, the two densities look really similar. Even though not quite, something here has changed in the relative order of these two peaks. And what they now suggest is that they should make something which is called dynamic time warping, where you now try to order the cells according to component 1. So I make a matrix where these are the control cells ordered by first CC, and these are the stimulated cells also ordered by first CC. And now for each element in this matrix, I now let's say these here in a couple of maybe 100 different bins, and these here in 100 different bins, so that each bin contains a couple of cells, and the value in each matrix element takes the correlation between these cells and those cells. The correlation calculated over a whole group of genes, not after the CCs, but over a correlation between stim and control cells over some genes. And for these some genes, we take genes which have a high influence on this first CC. And then once I have done this, give me one time walking. Beautiful time, very nice. Then what you then want to do is imagine it sort of turns out that this component here increases quickly and then slowly, and that one increases slowly and then quickly. Then I can now find the path of where I have the highest correlation, and ideally that would be along a diagonal. But in practice it can happen that it might look like this, where we might say, okay, this value in the stimulated cells seems to correspond to that value, and this value in the stimulated seems to correspond to that value. And what you then do is you make this matrix of correlations, and then you try to find a path through the thing from the top left corner to the bottom right corner that always follows through the values of the highest correlation. And that thing is called dynamic time warping, because originally it comes from the fact that from signal processing you have to record a number of processes, and they happen with different speed, and so you have to align of which phase belongs to which other phase. Yes, in dynamic time working you do a greedy search, where you always just go here till you found the maximum, and you go down a step, and you go on to the maximum, and so on. I think there's some reason why the greedy search usually works out reasonably well. And there's also different, I mean, it's sort of one of these things. You have this thing, and then you can probably look for algorithms to do that, and I'm sure there might be. As far as I remember, I always used the greedy search. I'm sure this will break down occasionally, and there's probably at least a couple of PhD theses which discuss about what you do in that case. But, so this is the step that I ignored now, simply because I didn't get that far. And with this, we can now do a and with this we can now adjust our coordinates such that they optimally fit together. And we do this for each of the canonical coordinates, and then we get the perfect integration. And that's a step I omitted now. I could get away with omitting it because we have this specific type where we know where the abundances of the cell types in the two conditions is the same, because it was just split, and the experimental time was not long enough for any cells to die, so that we know that all the cells which were present before are also present afterwards. But for more complicated things, you have to add this dynamic time warp afterwards. So now, if we now have this, let's look at a plot here. Let me see. Was given the gamma reduction is UMAP unintegrated, and my feature is this. Which one was that? ESC15. So now I can look for genes which differ between the two things. And here I show you one gene which is known to react strongly to interferon. I think simply stands for interferon stimulated gene number 15. And as you can see, in this cell type it reacts strongest. It goes from all gray to deep blue, and here it reacts a little bit less. What I could now do, I make this now not with the unintegrated, but with my integrated UMAP. Now these cells sit all on top of each other. At each place I get a mixture of an unstimulated and a stimulated cell. Probably to make this easier to see, I can use here this split by function, which allows me to split them like this. Yes, exactly. And now here, you see it has now split the UMAP using only the control cells to the left and the stimulated to the right, and I can directly compare for each places where does this, which cells, in which cells does the stimulation causes a strong increase of ISG15 and where does it cause a weak increase. And obviously what we would now need as the next thing is, first, we want a method to find many such genes. And second, we want a way to display this nicely. For the first part, how do we find many genes? An easy way would be to average the expression over each cluster. And in each cluster, we look at the difference between the average expression in the stimulated minus the average expression in the control cells. And then we try to find genes which show a strong difference in at least some of the clusters. And the other thing is we might now be interested, are the borders where the cells react, where the gene changes and where the genes doesn't change. These borders must not necessarily be along the cluster borders. Here they probably are, but maybe there's other things where we might say this is all gray and from here to here it's blue and here it isn't. And in order to do that, we would like to subtract these two pictures from each other, and to subtract and simply make a plot with one thing where we color by the difference. Now, if you want to subtract these two plots from each other, you notice an issue. What do I subtract from what? Here I have 5,500 control cells. Here I have 6,500 stimulated cells. I don't have an exact assignment of cell to cell here, because it's not even the same number of cells. So what I should better do is I make neighborhoods. For each cell here, I make a small neighborhood of maybe 20 or so cells around it. Maybe it's 20 nearest neighbor on this side, and it's 20 nearest neighbors on that side. I take the average over these two sides and subtract from them, subtract these two averages. And in that way, I can smooth out the individual differences from cell to cell. For example, here's a blue cell, and next to it is a gray cell. That's certainly not a real difference. That's just Poisson's. And I smooth out this thing by averaging over bigger sizes. And in that way, I then get a nice plot where I smooth, where I now get a smooth colored UMAP, which shows me where the gene differs strongly between stimulated and control, and where it doesn't differ so strongly. Now, how do we do that? I first thought I could prepare this and show you how to do this, simply doing and averaging over these cells and so on. Then I had a better idea, instead of doing it myself and make you do it. Remember, I always said I want at least those of you who want the grade, to do at least one little exercise. And I think I mentioned one possible exercise last time, and I felt a bit guilty because I only vaguely said it orally, and I can't even remember myself what exactly I demanded. Now you can use this to your advantage and show me anything, and if you say, yeah, this is what you demanded as a month ago, I'll probably believe you. Alternatively, you just can follow this slightly more worked out homework, where I now say, use this interferon D dataset where we've just done it, and let's first do the smoothing only for the control cells. So I do smoothing in PCA space, so I only use the control cells, do a PCA in the control cells, and so then I get for each cell, I get its 20 nearest neighbors, and then I can make a plot like this feature plot, which colors the cells according by the expression. I want to do that in a manner that I get rid of the salt and pepper thing, and that it looks smooth rather than suspect. And the way how I do that, I should probably put a link to this somewhere. I'll put it later when I'm back. So the way how I do that is, the first thing is, the easiest thing is, of course, I make a plot as it is here, color each cell by its own expression. The second possibility is, for each cell, I color the cell by the expression, by the average expression average over its 20 nearest neighbors. And when I do this averaging, I can do 2 ways. Either I use these log normalized values and average them, or I add up the counts of the 20 nearest neighbors, and I add up the totals of the 20 nearest neighbor, and then do the log normalization of that fraction. And probably the second one should be smoother and is also better. You might remember at nearly 2 months ago we talked about this when we talked about kernels moving along pseudotime trajectory where the Poisson distributed stuff. It's better to add up numerator and denominator separately and then divide rather than average over fractions. And then in that way, of course, you get two different plots. And you can then make these plots for a couple of different genes. Because we said we use only the control genes, this ISG 15 is probably not a good idea because it isn't expressed. Rather take one of those genes which we know where they differ between the different cell types. So for example, in this clustering vignette that we looked at the very beginning, you might remember where they tried to assign these different cell types here. Where was it? here, and in order to assign these cell types, they had here a couple of markers. So you can use these markers to do something, to have a few cells to plot this for. And then once you do that, once you have that done, of course you then can go back to our differential expression problem, where instead of smoothing over the neighbors according to PCA, we smooth over the neighbors according to CCA, and do this once for the stimulated neighbors and one for the control neighbors, and then we subtract them, and then we make this plot, which we don't color by absolute expression, but by the expression change between stimulated and control. And then we get this thing. So I put a lot of stuff, so I mean, it's probably if you get up to here, you've already done a nice homework. You go up to here, but if you feel motivated, you can look at a few more things. One thing we will have to look more probably later is these Pearson residuals, because we ask whether by the smoothing we might have over-smoothed. If you smooth over too many neighbors, you might average out differences which you would want to keep. And one way to do that is you ask the difference between my smooth expression strength and my actual count. Is this difference higher than what you would expect due to Poisson noise? And to do this, you ask what is the expected Poisson noise. And the expected Poisson noise is, of course, the Poisson variance is equal to the mean. So I take the square root of the mean to get the expected standard deviation. And what I then have here is simply the deviation of our observed value from the smoothed value put into proportion the expected standard deviation from the Poisson. So this tells us how many standard deviations we are out. And we can roughly say the usual thing, you shouldn't be out more than 2 standard deviations, shouldn't happen more than in 68% of the cases, you should be less than 2 standard deviations out, in 95 less than 3 standard deviations, something like this. So this is the idea behind Pearson residuals, that you divide your difference by the expected standard deviation, and then you look at a statistic of how often you see which values and ask, is this roughly standard normal or is it much wider than standard normal? In the latter case, you're probably over-smoothing. And yeah, I think the last part with the negative binomial, I just have to explain to you why we need a negative binomial here. So let's skip that for the moment. So now there's one more point. One thing that I haven't guessed here, I told you initially, these are different. These were 6 different donors. And now what would happen if there's a gene which reacts strongly to interferon in one of the donors, but not in the other 5 donors? Donor 1 has some strange genotype where his immune cells upregulate gene A whenever they see interferon. The others, this is not the case. In that case, what will happen is that we still see on average an upregulation. Of course, only 1/6 of it, but still it is. And we will report that this is one of the genes which go up by interferon. But we have now reported something which is idiosyncratic for one patient, as opposed to something which can be generalized. So we should check for statistical significance. And statistical significance, I always like to define it as something is statistically significant if you can be reasonably confident if you repeat the experiment with new material, like here, blood from new donors, whether will you see it again. And one way to solve this issue would be, at the moment I told you, in order to find genes that might be interesting between differentially expressed, do the following. Make a matrix with here the clusters as columns and the genes as rows, and each element is just the average expression of this gene in all the cells in this cluster. And do this matrix once using all stimulated cells and once using only the control cells. And then put the 2 matrices next to each other subtract them from each other, and check for each element in there whether there's a strong difference or not. Now if we want to do this properly, we don't do 2 matrices, but we do 12 matrices. 6 matrices for the control and 6 for the stimulated. Each of the 6 matrices has only the cells from one donor. And in that manner, I now have, for each gene in each cluster, I have a comparison of 6 control versus 6 stimulated. And then I do a paired t-test to compare these two. So I ask whether the average of the differences between stimulated and control is further away from 0 than the standard deviation of these differences, which, as you might remember, is essentially the logic behind a paired t-test. And in that way, I will only find genes which are, where the difference between stimulated and control cells is statistically significant in the sense that it's reproducible, that it happens over all, or at least over most subjects, and not only over one of our 6 subjects. Now, we are sort of at the point where we're now getting into where current methodology is currently under debate, because you might have noticed what I've just described to you assumes that our clustering has the right coarseness or fineness. If I have 2 fine clusters, then I might overlook stuff, because simply there is not enough cells in each cluster to get a robust average of the effect between stimulation and control. If I make my clustering too broad, then maybe I have a big cluster in which only a small parts of the cells react to the stimulus, with respect to the gene we're looking at, while most of the genes don't react, and hence we might overlook this gene. So I could now start in doing this with trying different resolutions of how fine my clustering is. But it shouldn't be necessarily. I can also look at what we've just done before, where we said, where each cell, where we just said we want to make this smooth plots, where for each cell we smooth over the neighborhood. And of course we can do the same. We can take, for each cell, take a neighborhood of its 30 nearest neighbors, and for these 30 nearest neighbors we do this whole t-test thing. 30 neighbors might not be enough, because we need a big enough neighborhood that we have a couple of cells from each donor. But we can do this, and in this way trying to get away from the clustering to a more smoother picture of finding regions in our latent feature space where specific genes react and other genes don't, in other regions where it might be the other way around. And how to do this in a proper way, that's something where there's currently a few papers written about it. Also doing this in such a way that we get a good balance between precise location in feature space as opposed to statistical power, because the more coarse-grained I have it, the more robust all my averages will be, because I average over more cells. So I gain statistical power, but I lose precision in what I can say in the end. So, but this whole smoothing thing will be, what I put here below as outlook is one more thing. You might notice that this smoothing here is smoothing simply over neighboring cells. Sounds nice, but it's a bit annoying, because sometimes the cell might be very dense and it should be easy to smooth. Sometimes the cells might spread out. The cells might be not very dense, and if I smooth over 20 nearest neighbors, there might be a few cells which are already completely different. And this smoothing sounds like the easiest way you can do. You could refine it a bit by not taking these 30 nearest neighbors all with the same weight, but putting a bit higher weights to the cells which are closer and lower weights to the cells which are further apart. But in all these cases, it's sort of not perfect. And one thing that people do a lot instead is to use a neural network and so-called auto encoder. And let's see if I manage till next time or the time afterwards, and then I show you this thing, which essentially means that we autoencoder, and who knows what an autoencoder is? Okay, quite a few. So the overall idea of the autoencoder is you have a neuronal network which takes all the expression of all the, you give it a cell with its expression vector, the expression of all the gene, and you ask the encoder part of the autoencoder to reduce this down to 20 numbers or so. Very much like in a PCA, you also reduce from all the cells to 20 principal components. And when you tell the decoder to invert this and go back to the original thing. And this is the same that conceptually our PCA is an autoencoder, because in the PCA we told it, take the whole thing, compress it down to only 30 principal components, and then he said from these principal components, if we undo the rotation, then we should get back the proper features. And we showed last time that the principal component is the linear operation which preserves most of the information in that way. But if we allow for nonlinear information, we can preserve much more information in our 20 dimensions than if we do it linear. So in that case, trying to in the PCA, the encoder and the decoder are just, the encoder is just a linear projection, and the decoder is just unrotating this projection. And we can show that as long as we are restricted to only using linear transformation, the PCA is the best we can do. But if we allow for nonlinear functions, we can do more. The autoencoder, the neuronal network, is a way to try to find the optimal function in much larger function space, and in that way improve this. And what will then, the hope behind the autoencoder is that what the neuronal network will essentially construct is a chart of our manifold. If we, for example, our manifold would turn out to be some 5-dimensional thing which is somehow strangely curved in space, like these bananas that I already always draw, then of course I might make one coordinate which goes along this, like our principal curves, another coordinate which goes across it, and in that case it is a chart of this manifold, and that's what when you try to construct the autoencoder, there's a specific kind called a variational autoencoder, where we use some mathematical tricks to force this thing to really converge towards a chart of a manifold, as opposed to some other way of representing the information. So, exactly, that's what I wanted to say for today. Let's see what we can do further, and yeah, it might be quite useful if at least those of you, if at least some of you would have a look at the smoothing, because getting a bit this feeling for the simple neighborhood smoothing is quite helpful to later understand what the autoencoder does. Also, this part, what I put here with the Poisson residuals, will turn out to be important to get the loss function of the autoencoder right. Our crucial idea will namely turn out to be that in PCA we said our objective is to minimize the squared error, so the Euclidean distance in our log normalized space. But with the autoencoder we will be able to not minimize, to not work in the log normalized space, to work in the original count space, and then properly minimize the Poisson likelihood. And yep, so I mentioned this now, you can already sort of get into your mind a bit, this idea that we'll have to understand Poisson likelihood in order to build a cost function out of it, and then we can try this in the next days. Okay, thanks, and see you next week, or possibly see you on, I don't know, will anybody turn up on Wednesday? I will probably sit around here and try to prepare next lecture. What I wanted to also mention is I will put on Moodle, I will open one of these discussion forums, because when you try this or the other exercises, what you will probably notice is that the difficult part is not getting the math into code, that's what we all know, but remembering all these little things of where do you find the PCA in its coordinates in a server object, And how do you get the neighborhood thing? And what was the name of this package again to find nearest neighbors? And yeah, it might help to simply have a forum where you can ask this, and either I or somebody else get with the answer, so that not all of you spend hours trying to figure out the same little basic steps, which I know by heart now after I've done it so often, but I know how much time I wasted in the beginning to look this up again and again and again. Okay, then see you.